Hi, in this video we are going to talk about quantitative PCR. You can find written notes on what we are going to discuss today at medicinecanbeeasy.com. The link is also in the description. So what are you going to learn in this video? I am going to explain what quantitative or real-time PCR is and how it works. We are also going to compare standard PCR and qPCR and discuss the applications of the method. Okay, so let's start. Quantitative PCR or real-time PCR is a modification of the standard PCR that has one important advantage. It allows us to monitor the amount of the amplified product in real time. When it comes to the components we need for the reaction, it is very similar to the standard PCR. We need a heat-stable DNA polymerase, primers, dNTPs, and the template. However, we use a special kind of thermal cycler that has a laser that illuminates PCR tubes. The DNA in the tube then emits a fluorescent light, which is captured by the detector, and the information is sent into a computer. The more fluorescence measured, the more DNA in the sample. That is the basic notion behind this method. Perhaps now you are asking, but how do we make the DNA sample emit a fluorescent light? Well, there are two commonly used methods how to do this. The first one uses a special dye that binds to the DNA. A common DNA binding dye is, for example, SYBR Green. If we take a closer look at the reaction, we will see that it starts with denaturation, when we separate the two DNA strands. In the second step, called annealing, primers anneal, attach to the DNA, and DNA polymerase is now capable of extending the DNA strand in the 5' to 3' direction during the third standard step called elongation or extension. The DNA binding dye then binds to the newly synthesized double-stranded DNA product, which, as a result, now emits fluorescent light. The more product there is, the more fluorescent light is emitted. The second method uses a dye-containing probe, for example a TaqMan probe. A probe is a short sequence of DNA that contains two important parts: a reporter that emits a fluorescent light and a quencher which blocks the reporter. This is important because we want the DNA to be able to emit fluorescent light after we have amplified the product, not before. Taking a closer look, you can see that the steps are once again very similar to the standard PCR. First, denaturation, when we once again separate the two strands, followed by annealing, when both the primers and the probes anneal to their complementary sequences, and now in extension the trick happens. As DNA polymerase synthesizes the new DNA strand, it cleaves the probe, which separates the quencher from the reporter, and as a result, the reporter starts emitting fluorescent light. And once again, the more DNA product we have, the more fluorescent light there is. Finally, we should talk about why this is all important. Why doesn't a simple standard PCR suffice? Well, as we already said, the key difference between standard PCR and quantitative PCR is that quantitative PCR gives us the information on how much DNA there is after each cycle, from which we can calculate the number of molecules in the original sample. In standard PCR, we only amplify the DNA sequence of our interest. We cannot do these calculations. So, for example, in diagnostics, when we need to determine the degree of infection, we can measure how much viral DNA there is using quantitative PCR. We can also use the technique to measure changes in gene expression by first transcribing RNA into DNA using reverse transcriptase. And there are of course many other applications of qPCR. So quantitative PCR is definitely something important that we should learn about because it really can be used in real life. Okay, so this is it. Thank you so much for watching. If you found this video helpful and you would like to watch more videos on genetics or medicine in general, please subscribe. Thank you again and see you soon.